I'm sorry about that. We are going to talk about chapter 12, patterns of inheritance. Sorry about that. Let's start with the father of inheritance, Gregor Mendel. Gregor Mendel was one of the initial scientists, or was the initial scientist who published a lot of studies about pea plants, which gave us the foundations of genetics and things that we have built on for the last 164 years or so. Kind of interesting. He was an Austrian Czech biologist, meteorologist, mathematician, and a monk. He had a lot going on. A lot of times back in these olden days, the only way that a person could, if you weren't of like born into a high class, the way that you could get an education was to become some kind of person of religious influence. And so we see a lot of people who were scientists were actually like monks and priests. And unfortunately for women, there weren't as many opportunities. And but you do see this in history around these like 17, 18, early 1900s. I just think that's kind of a interesting, people try and break the rules in ways that stay with the game. Okay, so Mendel just started very simplistically, which is such a great way in terms of science, is that you, regardless of how complex, how complex your study is, like for all of you, if you go on and you do research studies, even as complex as some of your research is, you will work with kind of basic things to begin with and then continue to build on those basics. And so what he studied, I mean, look at this, 30,000 pea plants. He took a look at very easy to study characteristics like what color, what height did they grow, what color were their, not only their pea pods, but their flowers as well. So smooth versus wrinkled seeds, very simple. He kept it very easy. In 1865, he published the results of his studies, and this was a lot of mind-blowing information because people didn't really know how did we get information from our parents in terms of what they looked like, how they acted, their internal physiology to us, like what happened there. And so he started really getting people thinking about inheritance and how inheritance comes from parents to offspring. And that also stimulated a lot of other people to figure out, well, what are these basics of inheritance? Like what are the things? And it got people thinking about reproduction and sperm and egg and how those might have been the things that passed on that we know today, passed on that information. Sometimes some of the things that he studied were he had plants self-fertilize. And now humans, we can't do that for a couple of reasons. We have some individuals that are born with both male and female reproductive systems, but that's a pretty rare thing. So to self-fertilize would be very difficult for humans because we just don't generally have that opportunity. But in many plants, they do have both male and female reproductive systems, and they can fertilize themselves. So he studied that to get some just kind of basic ideas about inheritance. And then he also studied when he took different plants, their sperm and egg, and combined them together, that what he formed were hybrids. And based on that, when we're talking about a hybrid just means like a piece of this and a piece of that come together. So when we're talking about hybrids in terms of sexual reproduction, we're talking about like some of the sperm information, some of the egg information, and they come together, which we know, all of us are hybrids of our biological parents as well. So let's talk about his first hypothesis. Now, in science, what we know is that sometimes we make hypotheses, and then after we do experiments, we find out that our conclusion does not support our initial hypothesis. And that's where Mendel started too. What Mendel first proposed was what's called the blending concept of inheritance. And his initial study, which, just like you all studied early in the semester, he used the scientific method. And what he found, I should say, in his scientific method, one of the things that he did was he did a little research, background information, and then he proposed a hypothesis. And his hypothesis said that if I had a parent who was, let's say, tall in terms of the pea plants and one that was short, that what the offspring would be a blend of those two traits, and so that all of the offspring would be medium, a blend of the tall and the short. So that's why we call it the blending concept of inheritance. But because what Mendel found still that the offspring were either tall or short, and there were no medium, he said, no, this study does not support my hypothesis. So, and my other example will be red and white, that his hypothesis would say that if you have a red plant and a white plant, that the red and white would blend together and come up with pink. And that's not what he found. He disproved or did not support his hypothesis by finding that he got red and white flowers still and no pink at all. So like a good scientist, he didn't give up, and he learned from that information, and he said, I'm going to make a new hypothesis and I'm going to try something else. So he started that scientific method over again. What he came up with that still holds true today is a kind of inheritance called complete dominance. And what complete dominance says is that when you have a trait, there are two different versions of that trait, or maybe more, and one version will be dominant over the other version or versions. And so that what we find is that a dominant trait will mask the presence of a recessive trait or recessive allele. And we're going to get into a lot of details of the terms as well. Okay, so let's think back to what we talked about on Monday. Why do you have two alleles for every trait? Let's remember, like two genes for every trait or two homologous chromosomes. Why do we have two of each of these things? Is it because one allele is located on each homologous chromosome? One allele is located, how's that sound? Pretty good? One allele is from your mother, one allele is from your father. That sounds pretty good. I see some head shaking. One allele is from the sperm and one is from the egg. What do you think? Yeah. Aha, yeah. So all three, A, B, and C, are, like you said, basically the same thing. So we're just using different, I didn't have it. So D is correct. All of the above are just the same ways of saying that why you have two alleles for every trait. So remember this. You have two alleles for every trait because one comes from egg, one comes from sperm, one comes from biological mom, one from biological dad. There's an allele on each of the two homologous chromosomes. So you have two alleles for every trait. You have two alleles for every trait. There are two alleles for every trait. There's two alleles for every trait. You have two alleles for every trait. If I'm not repeating myself too much, I have two alleles. We all have two alleles for every trait. So remember that, because sometimes when you get into doing problems in lab and then you start going like, why do I have this and this? Because you have two alleles for every trait. You have one from biological mom, one from biological dad. Okay. So a trait is controlled by two alleles for that trait. And those are found on homologous chromosomes. And you have two again because one comes from sperm, biological dad, one comes from egg, biological mom. Those traits are in a specific location on your homologous chromosomes. A location of a gene is called a gene loci. All of the different locations of genes are called gene loci. Locus, if we're specifically talking about one trait. So locus is singular of loci, plural. And then you could have alternate forms of a gene, like we just mentioned. Red and white are alternate versions of the trait for flower color, and we call those alleles. Those alternate versions are alleles. So below you have an illustration of a homologous pair of chromosomes. And in our homologous pair, we can have a number of different things. Here we have three gene loci. So as I have in the illustration here, you have loci pink, loci purple, and loci green, yellow. So let's talk about these. The top two, here you can see 2 pink and 2 purple, which means that this individual received the same alleles from both of their parents for both of these gene loci. When we get into talking about complete dominance, we're going to start to label those genes with letters. And what we're going to find is that if you have the two same for a trait, you will be capital A, or you'll be little a. And we'll get into maybe which one of these is which. But sometimes you can receive different alleles from your two parents. And if you receive different alleles from your two parents at a specific gene locus, you're going to be one letter and the other. You're going to have two different alleles to represent the different kinds of alleles at this locus on your homologous chromosomes. All right, so we are talking about complete dominance. Now, when you all, or if you all go on to take a genetics class, you're going to blow right past this complete dominance very quickly, because there are not a lot of traits that are found anymore in humans that are 2 alleles code for 1 trait. One that we still believe holds true in this in terms of complete dominance is handedness, is that that's one of the ones that we believe that there's just one gene loci, maybe this one here, that codes for your handedness, and you would get a capital R, 2 capital R's from each of your parents, or small r from each of your parents, or a capital R, small r. Most of our other traits, or pretty much all of our traits in our body, like when we're talking about traits again, we're talking about what we look like, how we function, and function can be your internal physiology, the workings of like your mitochondria, for example, or can be your behaviors. If you've ever noticed that, maybe if you know your biological parents, that your biological parents, one of them does a certain thing that you do pretty similar, or you act a certain way in reaction to some situations, that's inherited as well. So when we're talking about our alleles in terms of complete dominance, again, there's very few traits that are just like there's 2 alleles that code for that specific trait. Most of our traits are coded for by multiple alleles. Sometimes even hundreds of different genes code for 1 trait. And we'll get into that as we go further too. So this is the most simplistic way of looking or studying genetics. So when we're talking about complete dominance, you have a dominant allele and a recessive allele. The dominant allele hides or masks the presence of the recessive allele. Sometimes you don't even have a recessive allele around, and we'll get to that too. When we're talking about complete dominance, we use the same letter, uppercase and lowercase, for the trait. So if we're talking about handedness here, we're going to use R's to go with right-handed, which represents the dominant allele. So what we're seeing here is that we're going to use capital R for the allele for right-handedness, and we put it in uppercase. The recessive allele is there, it is present, but in the presence of a dominant allele, it doesn't express itself. So it's almost like it's hidden, or what we call masked. So I always like to think about it like this. If you are a person who is right-handed and you have a capital R and a little r, your capital R, your little r is there, it's in your homologous, it's on your homologous chromosome, but its expression is masked. And so even though you have a little r, you don't express it or show that in which hand you choose, or which hand you prefer, which you instinctively use, I should say. So your little r is there, but it is masked. So it's there, but not expressed. You carry it around with you, and when you have something like a capital R and a little r, you may pass that on to offspring, but you don't have, it doesn't have, this little r has nothing to do with your own handedness.
So you're saying it suggested that handedness is still two alleles complete dominance, right?
Yes, correct.
Codex, vein, ambidextrous.
Ambidextrous, so this is kind of, that's a great question. Ambidextrous people are typically genetically little r. They are actually genetically left-handed, but because our society, things are more geared toward right-handed people, because there's a very small percentage of left-handed people, that they are forced to use both hands. The other thing, there's evidence to support that. We'll talk about linked genes. If you remember when we talked about synapsis, that happens in prophase 1 of meiosis, that genes, you know, like if we're talking about a chromosome, and we have our sister chromatids, that this portion, let's say that that's going to do the crossing over, that when a chunk of a chromosome, when we look at it and what is found on that chromosome, that often, so what some people have hypothesized is that the handedness gene and one of the many forms of intelligence are very close together. And when genes are close together, we call them linked genes, that they are inherited together. And so that what we believe, what people have hypothesized, is that left-handed people have a certain kind of intelligence, and that because they have this, and it's like a upper intelligence in certain ways, that they are able to use their left hand and their right-handed kind of equally well for somebody who's ambidextrous, and it's linked to like a motor skill intelligence. So kind of interesting, but usually ambidextrous will be someone who is sorry, I'm using the wrong letters. Someone who is little r, or actually genetically left-handed. But this little maybe like boost in genes hypothetically could give them the motor skills to do things well, both right-handed and left-handed. In addition, we know that again, our society caters to right-handed people, and it forces them to get better at doing things right-handed as well. So good insight there. The left-handed allele, it's masked in the presence of the dominance. It is represented by the lowercase of what the dominant allele is designated as. So in this case, I know a lot of times we might go like, well, why don't we just use L for left-handed? But we're going to keep our letters consistent. And so we said that dominant is capital R, and we're going to use a little r for the recessive form of this complete dominance trait. All right, so again, oh, look, 2 alleles for every trait. There's 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 2 alleles for every trait. You get those 2 alleles from your biological mom and your biological dad. You get them from sperm and egg. So you have 2 alleles for every trait. You have 2 homologous chromosomes. There's 2 alleles for every trait. Something to remember. All right, so our choices of combinations for our alleles for handedness will be a capital R from biological mom, a capital R from biological dad, or a capital R from biological mom, a little r from biological dad, or a little r from biological mom and a capital R from biological dad, or a little r from biological mom and a little r from biological dad. So we have those 3 possible combinations. Just to point out to you, these are the same thing. So let's say that in this case, you have 2 scenarios for your capital R, or little r, capital R. In one case, your biological mother could give you the little r, but in another case, maybe she can give you the capital R. So these 2 mean the same thing. What is really good to do is to stick with these combinations here of capital R, put the capital letter first and the recessive letter second when we're talking about this combination, because your brain does something funny sometimes. When you write something like this, because of stress and especially in summer, you're so accelerated, you have extra stress on taking this class. Some of you on an exam might look at this and go, oh, this is recessive because this is going to mask that. But that is not true, right? Regardless, if the dominant allele is in your second position, it's still going to mask this one. And so just to help yourself visually connecting your eyes to your brain appropriately, even if it's, you know your biological mom and you always put the mom first for whatever reason, if she gives you the recessive letter, try and put it after the dominant so that you minimize those kind of, silly, stupid mistakes of going that your brain looks at this and goes, that masks that, when that's not true. So this helps you in organization to just keep it straight in your head that this will mask that. It's one of those things that I've seen a lot of students who are resistant to always putting the dominant before the recessive. They get things wrong on the exam and go, yeah, I should have listened to you. And it is, again, just like your brain just pulls off something that is not to be true. Okay, so let's talk about when we have names for the different pairings of alleles. So let's start out talking about what we call homozygous dominant. Remember, we had homologous chromosomes. They both have HOMO in there, which we know means similar or same. Homozygous, similar or same. Dominant means that you have two of the same dominant alleles. So homozygous dominant, if you just told me, I said, what's your results for this Punnett squared that you did? And you might say, I have two homozygous dominant, two heterozygous, no homozygous recessive. I know what you mean is you have two capital R, two capital R, little r, and no little r, little r. And so we can use those as like translational words. Okay, so homozygous, two of the same. Dominant, two of the same dominant. We call that a homozygous dominant trait, or homozygous dominant genotype. And when the way this is expressed, there is no recessive. You have a dominant, so it is going to be expressed as the dominant trait, which is right-handed. Now, we could have two of the same, but recessives, and you have two of the same homozygous, but they are recessive, so you will be little r, little r. When you do not have a dominant allele, it's going to be expressed as the recessive trait. So in this case, that person will be left-handed. A homozygous person can only pass on either, if you're homozygous dominant, capital R's. If you're homozygous recessive, only little r's. You can only pass on the genes that you have. So when you're talking about homozygous dominant, and let's say that we're talking about a female, that the homozygous dominant female, she could pass on a big R or a little r. Thank you. Thanks so much. I'm sorry, they're in the way. When somebody is homozygous recessive, they have little r in their eggs that they form. Let's say it's a biological male. In the sperm that they form, they can pass on in each of their gametes a little r or a little r. So in this case, what I'm talking about is when they're passing on their alleles. These are formed in meiosis. And what we're talking about are either sperm or egg. All right, so again, homozygous parent who's capital A, this person is homozygous dominant. When they make their gametes, it's kind of like you can just split this in half, right? And when they make their gametes, they're going to have actually four. If we go through all the stages of meiosis, they would have four capital A gametes. Just like this. If you have an individual who's homozygous dominant and the dominant allele codes for purple, a big P individual will have sperm or egg that are big Ps. A recessive parent, which codes for little p codes for white, the recessive trait in this case, they can only pass on white in their gametes, white genes in their gametes or white alleles. All right, so what if you have one of each? If you have a capital R and a small r, like my example up here, that person is what we call heterozygous. Hetero, in our society, if you're heterosexual, you date someone of the other sex. So hetero means other. You've got one of each kind, or two different kinds. Just like I said here, if you are somebody who's capital R, same thing here, the dominant allele will mask the recessive. So if I have a capital R and a little r, I am going to be right-handed. I'm not half right-handed because I have this. This is totally masked and hidden. So I am as right-handed as somebody who is capital R, capital R. That's the way complete dominance works, is that either you are the dominant trait or you are the recessive trait. There is no blending. Remember that Mendel, he kind of said, no, this doesn't come out to be right. And so you are capital R, little r, your capital R is going to mask your recessive little r, and you are going to be as right-handed as someone who is big R, big R. This individual can pass on either capital R or little r alleles in their gametes. So here again, you have a big A, little a. They can make gametes that are capital A allele or little a allele for this trait. And here again, this is, I like this one a lot, this illustration, because we go back to, if you are an individual who has received a capital P from your biological father and a little p from your biological mother, you are as purple as someone who is big P, big P. And then same thing here. Let's say that the biological father gives the little p for white and the big P for purple. The purple is going to mask the presence of the white. And again, you're as purple as somebody who is big P, big P. So Mendel created what are called monohybrid crosses. Mono means one. So what we mean by that is he is going to look at one trait at a time. Mono, one. Hybrid, we're going to take two different parents. We're going to take those together and make a hybrid or a cross of genes in our offspring between two different parents. He started out very simply, because again, I said he was a good scientist, right? Science, we like to keep as simple as possible to start. So what he started with are what are called true breeding, or sometimes we refer to it as pure breeding. And what that means is that both alleles are the same in each parent.
How would he know that both alleles are the same?
He would have had to have done a number of crosses over the generations to see if ever a offspring in many generations came up a different allele or a different expression of the trait. So he has to ensure that he has a parent who's capital R, he may have had 10 generations of crosses of purple plants that he kind of filters out anybody who might be carrying a recessive allele. Really good question. And then white is probably white is definitely a lot easier because white can only have little r and little r in there. But it would take some work to ensure you have a capital R parent. So when we're talking about this kind of cross, again, we call it a mono, one, hybrid, crossing two different parents to get, or just crossing parents in general. You're a hybrid of your biological parents because you get some of each of their genes in you. But he did this with true breeding or pure breeding parents. So we're going to go to capital T, capital T. And we're going to use our T's for our trait, and we're going to look at height. So he took a homozygous dominant. It's pure breeding because it's two capital T's. He crossed it with a homozygous recessive, two little t's. And he predicted what would happen by mapping this out with a math or a functional method called Punnett squares. All right, the Punnett square. Again, simple calculation method to see what kind of genes might our offspring inherit. Like I said, capital T is the allele that codes for tall, little t is the allele that codes for dwarf or short. So he starts out with a capital T and a capital T and a little t, little t. Okay, so just to remind you of how gametes are made, if we have an individual who is capital T, let's walk them through the process of meiosis 1 and 2 to ensure that we get big T's in our single big T in each of our gametes. So this individual, remember that prior to cell division, if we're talking about any kind of cell division though, the DNA has to replicate. So before we even get into meiosis, the DNA has to replicate, and what we get is you get each of our original parent alleles, and they each get a genetically identical sister chromatid. So as a result of the DNA replication that happens before meiosis, you get the sister chromatids form. And what I'm showing you here is that we have a capital T that is connected by that nucleosome or that centrosome area. They are temporarily connected as two sister chromatids. Then these are going into meiosis 1. Remember that during meiosis 1, that you get two means by which we get genetic new combinations. One is through crossing over in prophase 1, and the other one is independent assortment in anaphase 1. Did you want to? Okay. All right, so we get our two cells. So remember, these are in one cell. So we go from our DNA replicating, we get one cell. End of meiosis 1, you get two daughter cells. Then meiosis 2 occurs in both of these daughter cells. And then what happens is that you have the separation of the sister chromatids in meiosis 2. And we get our gametes. And we have 4 haploid gametes. Remember that this is diploid. And when we take a look at our numbers, whether they are diploid or haploid, this one is diploid, 2n number. This is diploid times 2, so we're essentially saying 2n times 2, meaning that each of our original homologous chromosomes get a paired sister chromatid. So that's why we say 2n times 2. These technically, what we call them is we call them haploid, but times 2. Because the sisters are still connected, we count them And this is a thing. Paired sister chromatids, when they're together, they're called one chromosome times two. Something to wrap your brain around. We call this an N times two. So this is N times two. And this 2N refers to our original homologous pair times 2 times 2. If any of you go into like genetics or you go into cellular engineering or genetic engineering or one of those fields, genomics, this is a very kind of like they like to call a sister chromatid pair one chromosome times 2, just so you know. And if that comes up again in like a further class, then you know that too. And when your sister chromatids, when they separate at their centromeres, then you get your haploid, or N, in number cells. And we could do the same thing for our little t, and our result would be 2 little t's, blah, 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 with all little t gametes. Anyone have any questions on that? I know sometimes in biology, it's like sometimes things are just like real easy, like diploid too, right? Di means 2, and then sometimes this stuff gets a little more picky and precise. Okay, so let's calculate out our genotype and phenotype. What do we mean by genotype and phenotype? Genotypes are the genes. What are the letters that represent the genes? Big T, big T, big T, little t, or little t, little t. That's what we call a genotype. A genotype is going to have 2 alleles for every trait, because you got one from biological mom and one from biological dad. The phenotype is the physical. See, PH, pheno, PH, physical. So the phenotype is the physical expression of the genotypes. So I can go here when I want to know, like, what are the phenotypes for these genotypes? And I would say these 2 are tall. That's their phenotype. And little t would be dwarf or short. So we're describing, again, the physical expression of the genotypes is the phenotype. Typically, when we're doing Punnett squares, you are going to be asked, what's the genotypic ratio? How many of these to how many of these? How many of these do you have? And then what's the phenotypic ratio? How many of the dominant expression? How many of the recessive expression? We always keep them when we're talking about these genotypic and phenotypic or genotype-phenotype ratios. We always keep them in the order of homozygous dominant, heterozygous, then homozygous recessive. So if you said to me, I've got a 1 to 2 to 1 genotypic ratio, I would know what you meant by 1 to 2 to 1 is that you got 1 homozygous dominant, capital T, 2 heterozygous, big T, little t, and 1 homozygous recessive. We always keep them, again, in the order of big T, big T, big T, little t, little t, little t. When we're talking about the phenotypic ratio, you always put the dominant trait first and the recessive trait second. So if you said I got a 2 to 2, I know it's 2 tall, 2 short. If you said I got a 4 to 0, I know it's 4 tall, 0 short. If you said I got a 0 to 4, I would know that you had 0 tall, 4 short. All right, so Mendel, again, he's real good, real organized. So he tracked the generations by calling them different names. The individuals he started with, he called them P for parent generation. F1 is either refers to the first generation, or sometimes like you'll read it in a book and it'll say first familial generation, F for family, familial. F1 are the first round of kids. F2 are the second round, meaning the F1's kids. So think about it as parents, kids, grandkids. we track those generations, right? Just we use different names. Okay, so we're going to go through an example where we cross two different true breeding. True breeding means you have two of the same but different parents. We have a true breeding capital P, and a true breeding little p, little p. So what we're going to do, we're going to make a box. And why did I do 4? Because I have 2 alleles for one parent, and I have 2 alleles for the other parent. If you're someone like me who often writes fast and then it gets messy, this is where you need to slow down and be very careful and exaggerate your letters, because P's big and little look very similar to each other. So I like to be I like to exaggerate a lot and show that I've got one parent who's the big P and one parent who are the 2 little P's. That way I know a little tiny p, it is showing the recessive trait. In genetics, typically males go on the side and females go across the top. So in the future, if you're doing a genetics class, it's always males on the side, females go across the top. Males are, females are. This is the male symbol, this is the female symbol. Now what we're going to do is this is a P generation and a P generation. Oh, geez, dangerous. Inside, all of these are going to be F1 generation. Now, when we do this cross, what we are going to do is we are going to take this capital P, we're going to put it in the boxes across. We're going to take our second row of capital P, put it in the boxes across. We're done with this parent. We're going to go to the top. Our little p is going to go in the 2 boxes straight down. And our second allele for our second parent, little p, the boxes down. Now we're going to take this information in here from our F1 generation of offspring, and we're going to go here and go, okay, how many big P offspring do we have? None. So we got a 0, big P. How many big P, little p? 1, 2, 3, 4. 4 big P, little p, and how many little p offspring? None. So our genotypic ratio for our F1 offspring are 0 to 4 to 0. Then we're going to take that, we're going to translate how many of them appear to be purple, how many appear to be white. Remember that these 2 genotypes code for purple, and this genotype codes for a white appearance. So we add these up, 4 and 0. That's our first generation. Let's say that we're working with a type of purple plant that can fertilize itself, so that we can just take any 2 of these. Well, they're all the same. So we pick 2. They self-fertilize. So you take a sperm and an egg from your F1 generation, and we fertilize them together. Oh, wait, before we get there. So here again, you're showing that remember that this individual was big P as their genotype. This individual, little p, they're both true breeding. They both have 2 of the same alleles. And what they made here are big P, little p offspring. Okay, so now we're going to change this up just a little bit. We're going to do our second generation. So one of our parents goes on the top, big P, little p. The other one goes on the side. We're going to put the big P across. Now, when we get to this row, I have a suggestion. So remember, your brain can play tricks on you. What I'd like to do to minimize mistakes is think about there's 2 alleles going to be in each of these boxes. This one's a little p. Instead of putting it here, just in case a dominant allele will come along, what I typically do with a recessive allele is I put it in the back. Because again, a little p, big P is the same thing as a big P, little p. So just in case something comes along that's a big P, which in this case here, when I put the other parent, the female parent's alleles down, I do get that big P, little p. And it just mentally looks cleaner, nicer, limits my mistakes. And then this second allele for our female parent, put the little p down and a little p down. These, remember, they're our second family, familial generation. So each of these is an F2. Our parents were F1. So now I'm going to take a look at my genotypic ratio, and I have one of these, one big P, 2 heterozygous, 2 big P, little p, and 1 recessive, 1 little p. I'm going to translate my genotypes into phenotypes and know that anybody who is big P or big P, little p will be expressed as purple or show the purple color or trait. And then my little p will be white. So in this case, we finally do get a white offspring in our third generation. And I say third generation because we had parents, F1, F2. All right, so that's like our basics of our Punnett squares. And then here, that our big P, little p self-fertilized, our F1 generation to make our F2. And 3/4 or 3 out of 4 were purple, but remember this one was big P. These 2, heterozygous, big P, little p. And our white has the genotype of little, let's make this like really tiny, of little p. We could also look at this on a Punnett square. If we put the actual flower colors in, and you could see that your heterozygous parents made the F2s of big P, little p, 2 of those, and 1 little p. Anybody, any questions? Okay, I want you at your desk, we're going to try out practicing some of these Punnett squares. So go ahead And feel free to check in if there's somebody around you that you want to. You don't have to do that. But go ahead and do this Punnett square. Draw out the Punnett square for lab. I know some of you are just like, I got this. Some of you can look at this and go, I know what the genotypic and phenotypic ratios are without even doing the Punnett square, but for lab, you are going to have to draw out your Punnett squares regardless. So do your Punnett squares, draw the whole thing out. And I know for some of you, this is like, oh, this is like, I get it, okay? But we're building, so remember, we're building on this. I appreciate your patience. Good reminders of these like little details of Punnett squares. So I'm going to start writing out the way I would do this Punnett square. Okay, so what's the correct answer? A, B, C, D, or E? Okay, so this shows you that when we do the Punnett square, you get two capital P, and two little p, two big P, little p. Okay, what's the phenotype ratio? So now we're going to translate that into our purple. So what's the answer? Very good, for purple. Remember that these, the ones that have the dominant allele, at least one dominant allele, code for purple. So we've got 4 of those, and we have no white. So A is correct. Okay, let's do, oh, sorry, yeah. Right, what the parents were, yes, and we're going to get into that too. When we go backward, we call it a test cross. But yes, good question. We'll do that. Why don't you all take a break, and then when you come back, we'll go through that last question on the page. So come back at 10 after.